Hello and welcome back everyone. Today we'll be discussing about epigenetic marks. Okay. Now in the previous class I told you that human body is made up of trillions of cells, right? All those cells differ from each other even though they have same set of DNA. That's what we discussed in previous class. Why do they differ even after having same DNA? Because DNA is like script, right? And depending on who is reading the script, the outcome differs. Okay. So this explain why, but this doesn't explain how the process happens and how DNA is read differently by different types of cells. So the DNA is read differently by different types of cells due to epigenetic marks. Due to epigenetic marks. So what are epigenetic marks? Epigenetic marks are chemical modifications on DNA or histones which are caused over a period of time due to environmental factors or due to some other factors. Okay. Now, an interesting fact is that epigenetic marks can be passed down from parents to children. Okay. So these are the chemical modifications on DNA or histones. What are the histones? Histones are the proteins which helps DNA package, which helps in DNA packaging. Okay. Like DNA is wrapped around those DNA is wrapped around those uh, proteins and it is stored in the cell. Okay, so the chemical modifications on DNA and histones are what we call. So epigenetic marks can be of different type. I'll be discussing about what are different type of epigenetic marks in upcoming videos. So it can be methylation, it can be acetylation, it can be phosphorylation, somylation, ubiquitination. Means methylation means, as the term suggests, addition of methyl group. Acetylation means addition of acetyl group. Phosphorylation, addition of phosphoryl group. So these are the type. These modifications are called as epigenetic marks, which helps cell to read the DNA in different manner. Okay, it differs from cell to cell. So epigenetic marks are of two types. One, repressive marks. As the name suggests, repressive means repression. Okay, so this results in gene silencing. Okay, it means if the epigenetic mark is added on the DNA, that gene will be silenced. Okay, so this is the DNA and I add epigenetic mark here, and this is a repressive mark. Okay, so when the gene will be, so this is the gene, and this from here. Okay, I'm sorry. This is so from here the machinery comes. The DNA reading machinery comes and start transcribing the DNA. But oh yeah, there's a gene silencing the repressive mark. So this gene will not be repressed. This gene will not be transcribed. So this repression of gene is due to different factor. It could be due to direct perturbation of the structure of the chromatin structure, or it could be due to recruitment of the repressor or recruitment of the proteins which help in gene silencing. Other type of marks are active marks. Again, as the name suggests, that those help in gene expression. Okay, so if there's an epigenetic mark on the before the gene, it states that this gene should be activated. Again, it could be by direct perturbation of chromatin structure, or it could be by recruiting the chromatin remodelers. I'll explain how does this happen in later videos. But just for now, take into account that epigenetic marks are of two types: repressive marks and active marks. Repressive marks, if there is repressive mark on the DNA or on the histones, it will result in gene silencing. And if there is active mark, active means go, repressive means stop. If there is active mark, it will result in the gene expression. Okay. So this epigenetic marks, for understanding it better, imagine epigenetic marks are like a punctuation marks. Okay, so there's a statement here. Okay, can you read this statement now? No, it's tough to read this, right? It's just a scramble of words. So for reading this, what you'll have to do? You'll have to format it first. Enjoy. You'll put a space here. The. You'll put a space, then again present, you'll put a space here, moment. Okay, you'll put a space here and here. Okay. Now again this somewhat makes sense, but we don't know whether this is a full sentence or this sentence will be in continuation. So for making sure that the sentence end here, you have to put a punctuation mark, right? You'll put a full stop here. But now you want to say that there's a small gap, so enjoy the present moment, dear. So for the small gap, you again put a punctuation mark here. So there's start. This is the start of the sentence. Then you start saying the sentence. You take a little pause here, and then the sentence ends. So this is what punctuation mark does. That punctuation marks helps to demarcate the sentence boundary. Okay, it tells us that here from here the sentence starts and here it ends. So punctuation, so epigenetic marks are kind of punctuation marks for reading the DNA. Okay, now I hope you get that. Okay, so for understanding epigenetics, for understanding the molecular mechanism of epigenetics, you have to have knowledge of chromatin. Okay. Now what is chromatin? DNA is not uh, packed in the cells as it is. Okay, and this is the cell, this is the nucleus, and in the nucleus there will be DNA. But what do you think? That DNA is just lying around in the cell? No, it is actually packaged in the cell. DNA is 2 meter long. Okay, this each cell has 2 meter long DNA, and that is packaged in around 66 micrometer of the cell. So how does it packaged? It has to be wrapped around proteins which are known as histone proteins. These are positively charged proteins, and on those proteins, DNA is wrapped like a string, and it forms a structure which we call as beads on the string. This is called as beads on the string structure, and this beads on the string structure is referred as chromatin. Okay. So for understanding molecular mechanism of epigenetics, we have to have knowledge of chromatin. So this is DNA. So just take a note that DNA plus histone, this is chromatin. Okay. So the chromatin shows different compaction in different regions of chromosome. So what is chromosome? Chromosome is highly dense, like highly dense structure which can be seen during cell division. Okay, this can be seen during cell division. So this highly dense structure, what is it made up of? Of course, it is made up of chromatin. Chromatin is packed in a higher level of order, and it forms chromosome. So in the chromosome, in the different region of chromosome, the compaction of chromatin differs. Like it will be densely packed in this region. It will be densely packed in this region. This is called centromere. So in the centromeric region, it will be densely packed. In the telomeric region, it will be densely packed. But in the distal arm, this is the distal arm, it will be loosely packed. So depending on the degree of compaction, tightly packed or loosely packed, we call them heterochromatin and euchromatin. Okay. So the densely packed structure is known as heterochromatin, while loosely packed structure is called as euchromatin. So whenever there's staining of chromosomes, the dense stained is heterochromatin, while the faint stained, lightly stained is euchromatin. Okay, so this heterochromatin, because of it is densely packed, it is transcriptionally inactive. Means though the genes in the heterochromatin are not expressed. Okay, those are silenced. While the euchromatin is transcriptionally active chromatin. Okay, so these genes are getting transcribed continuously. The euchromatin are so the euchromatin consists of the genes, housekeeping genes, which are always needed by the cell, like ribosomal genes. Okay. And well, heterochromatin is consisted of telomere and centromere, which is kind of structural part, which is not needed, which is not needed for like it we don't require the transcription of that part. That part consists in heterochromatin. Okay, here are some differences between euchromatin and heterochromatin. So euchromatin, as I said, it is lightly stained. We call it decondensed. In the interphase, it appears decondensed. Interphase means the when cell is not dividing. Okay, it is that phase of cell cycle. Okay, so in the decondensed, if you see some lightly stained structure, it is for sure it is 100% euchromatin, and if it is highly dense, it is heterochromatin. The chromosomal location, as I said, on the distal arms, it is euchromatin, while on the pericentrometric region, means the centromeric region and telomeric region, it is heterochromatin. Replication time. Okay, so now tell me, what do you think that what should be the replication timing of the euchromatin and heterochromatin? So euchromatin will be transcribed throughout the space. Why throughout the space? Because all those are very important gene, and it will be replicating throughout the replication phase. S phase is the phase where the DNA replication happens. Okay, the cell prepares for a DNA replication. Well, the heterochromatin, which is structurally, it is very dense, right? You have to uncoil it first, and then you'll have to replicate it. That's why the replication happens in late S phase. Okay, sequence composition. Euchromatin has unique DNA and middle repetitious sequences, while the heterochromatin have repeats. It has repeat regions. Okay, repeat regions. This is a very important point, that it has repeat regions. This is transcriptionally inactive DNA. Gene density is variable in euchromatin and it's low in heterochromatin. Nuclear location is dispersed of the euchromatin, while in heterochromatin it is often clumped due to high degree of compaction.